In every organism on Earth, proteins are essential for survival. They catalyze reactions, regulate the cell, fight off bacteria, and are responsible for a myriad of other processes. But the really interesting part about proteins is the similarity seen between organisms. For example, the protein keratin is found in our fingernails, dog's fur, and birds' feathers. And the reason for this is because the instructions for making proteins have been passed on for generations in the form of DNA. In eukaryotes, DNA is stored in the nucleus in the form of chromosomes. Chromosomes are repeatedly wound strands of DNA. This tight structure can be unwound to allow the cell to access coding segments of DNA known as genes. Genes are comprised of a code in the form of base pairings that give instructions for building proteins. Before moving on, I just wanted to talk about the structure of DNA itself. And the really important thing to take away from this is the complementary base pairings. As you can see, A's and T's always bond with each other, and the same for G's and C's. So what you can do is you can look at one side of the molecule and you can predict what the other side would look like. And it's also important to note here these 3' and 5'. They are just saying that this molecule runs anti-parallel between its two sides. And that's just talking about the orientation of its sugars and phosphate groups. RNA is a very similar molecule. It's a single helix, unlike DNA, and instead of thymine, it has uracil, but it can transfer that information from DNA and move it throughout the cell. So just an overview, we have genes that are on DNA, and then we can take these bases on the DNA, translate them into RNA here, and then we can transfer that information to make proteins. Given the fact that DNA is in the nucleus and protein-producing ribosomes are in the cytoplasm, DNA's info needs to be transported. This is done with a molecule called messenger RNA, and its formation is called transcription. The starting point of transcription is marked by a segment of DNA called the promoter. Promoters are also called Tata boxes due to their base pairings. The molecule that produces the mRNA strand is called RNA polymerase. RNA polymerase binds to the promoter with help of proteins called transcription factors. The combination of these molecules is called the transcription initiation complex. Once RNA polymerase is bound to the DNA, it pries open the double helix and adds complementary nucleotides to the template DNA strand. Eventually, RNA polymerase will code for a polyadenylation signal, or AAU-AAA. This marks the end of the transcript and releases the mRNA. In eukaryotes, mRNA needs to be modified before leaving the nucleus. This process is called RNA processing. First, the 5' end of the RNA molecule receives a 5' cap, a modified form of guanine. After transcription, the 3' end receives many adenine bases, forming a poly-A tail. These additions facilitate the exportation of mRNA from the nucleus, help protect it from degradation, and help ribosomes connect to the strand. In addition to the modifications on the ends of the RNA, complexes called spliceosomes remove segments of non-coding bases called introns. The remaining coding segments are called exons and are joined together by the spliceosomes. Once these changes have been made, the mRNA can travel to the cytoplasm. The following process is called translation because the genetic code is translated from nucleotides to amino acids. First, a ribosomal subunit attaches to the mRNA. Each group of 3 nucleotides in the mRNA is called a codon and codes for a specific amino acid as shown in the table. Once the mRNA is attached, complexes called transfer RNA can match the base pairings in the codon to their complementary anticodons. The tRNA also carries each codon's respective amino acid. For example, the start codon in translation is AUG, which codes for methionine. Once the initiator tRNA attaches to the mRNA, a molecule of GTP is expended and the large ribosomal subunit attaches with the help of proteins called initiation factors. The combination of these structures is called the translation initiation complex. In the large ribosomal subunit, there are 2 distinct chambers, the P site and the A site. tRNA molecules bind to the A site and bring the next amino acid in the chain. The preceding amino acid then binds to the new amino acid, and the previous tRNA molecule can exit the ribosome. The polypeptide elongates as the tRNA and mRNA molecules move through the complex. Each tRNA shift causes 1 GTP molecule to be expended. Eventually, the mRNA will code for a stop codon. Stop codons bring proteins called release factors that hydrolyze and release the polypeptide chain. The use of 2 GTP molecules can then break down the complex. It's important to note that the ribosomal subunits are reusable and the tRNAs can pick up more amino acids with the help of enzymes called aminoacyl tRNA synthases.
In short, proteins are pretty cool and allow us to do a lot of pretty cool processes, such as growing, cell-to-cell recognition, and of course the coolest process of all, getting rid of the daily quizzes.